Okay, we'll go ahead and get started. Well, hi everybody.
you.
I am Chou and I am in Colorado. So I'm 2 hours behind Eastern time, whatever that is. And snow was here last week. So today's a beautiful day in Colorado. And I teach in Longmont, Colorado, and AP Biology and Biotechnology. So these kits that we're going to talk about today, I actually used with my students yesterday. So I'm very fresh off of using this with real life students. So hopefully we'll have a fun time together today. In the chat, as you join us, go ahead and type in where you're teaching at, what kind of situation are you in? Are you middle school, high school, or college? And what kind of courses do you teach? So I'm seeing all across the place, Pennsylvania and Washington, so coast to coast here. Variety of schools. And I guess the question to ask is, who's actually seeing in-person live students and who's all virtual? And for where I'm at, we started our semester with all virtual and have moved to, and then did hybrid. And now I think next week we're back to virtual. So it's a ping pong. So today we're going to talk about models, using models to make connections between DNA and chromosomes. And hopefully many of you had pre-signed up already to receive this kit. If not, no worries. Our 3D Molecular Designs is happy to send you a free kit so you can sample this and do it for yourself at home. So some of the things we're going to do is model chromosome structure from the DNA nucleotide level all the way out to the condensed chromosome. And we're also going to talk about genetic variation as we model mitosis and meiosis, and throw in at the very end a connection with using this particular kit to construct Punnett squares and connect at the genetic DNA level to what happens phenotypically. All right, so let's go ahead and get started. So in my classroom in typical years when I see students, we use a variety of these 3D molecular design kits. And you can see these are my students actually from last year, pre-quarantine, and from anything from DNA to modeling processes such as osmosis and replication, transcription, translation. And so this kit that we're using today is one of my favorites because it's unlike some of the other ones out there where we can actually model crossing over happening between between homologous chromosomes. So, and we're going to be in your hands if you pre-signed up for this kit. We're going to be working with a student modeling kit, but the next slide shows in the classroom. You can buy the classroom versions where I have usually purchased 6 of them with groups of like 4 to 6 students working together. So this was last fall as we modeled. You can see there's a little bit of crossing over happening. So that was meiosis. And in terms of management of what materials to provide students in the classroom, it's I have these trays and have them all set out. So we're going to take a look at the student version of this kit in the next slide. And these are some of the components that are part of it. And as for those of you who have this kit in your hands at your house, in the next slide, this slide, you'll see that there's a couple of cardstock pieces that come with this kit. The scaling graphics, and you'll need to do a little bit of arts and crafts and cut apart these graphics with scissors, a little bit of tape as well to assemble the Punnett squares, and some glue to glue on the foam pieces. You're going to see in my camera here, so that you glue it to the cardboard pieces right here. Okay. But you don't need to do that for this workshop together. We're going to go ahead and take a look at the foam parts. So on the next slide, you'll see what I would like you to do is, if you have the graphics, you can just kind of lay it out. You don't need to take the time to cut it apart with scissors right now. Maybe do it later at home. But go ahead and assemble these chromosomes shown here. And so you're going to see that there's going to be parts of it that are larger foam segments. Those are going to represent the condensed chromosomes like you're seeing on the left, a middle scaling graphic. And what's nice about that is, we'll talk about in the next slide, is your students can see visually the connections from when you switch from saying DNA sequence and the nucleotide sequence all the way out to nucleosomes and chromatin to condensed chromosomes. So I'm going to pause for a little bit for you to be able to construct those. And the middle sequence that you're seeing in that picture previously is, don't worry about the identical sequence, just put a few nucleotides in there, of course matching A's with T's and C's with G's to be able to show that complementarity of DNA. So in the classroom, students would kind of start with this basic model of chromosomes, and then I will show them the next slide in that graphic, and that will be the organization of eukaryotic chromosomes. So this is the graphic I show, and then in the next slide, you'll see that the students will put their chromosome in front of them and then compare it to the slide, and we go through and talk about how zooming in really close, you see DNA is a double helix made up of nucleotides that are paired. And so that's so Heather, if you could, or circle that area, yep. And then as DNA is wrapped around histones, those are the proteins that help to organize all this long, windy DNA, and that's part of the scaling graphic. And as we zoom out, this chromatin is organized into loops, further condensed, and then when cells are getting ready to divide, that chromatin condenses into visible structures called chromosomes. And so from the nucleotide level, zoomed all the way out to the visible chromosome level, students have the ability to see that through this model. Okay, now let's go zoom out further. One comparison I want to make is that the classroom kit, which is on the left, versus the student modeling pack, which is on the right. In the classroom, that large classroom kit has all blue nucleotides or all red ones that you can use to show paternal versus maternal chromosomes. The student version has the colorful ones with blue, red, green, yellow, and these same nucleotides can be used for other things like DNA replication, and so it's kind of handy to have that. So for all of my students this year, because we started virtual, I purchased like 54 of these student modeling packs for my 54 students in AP Bio this year, and it's been really helpful, especially since we've bounced back and forth between hybrid and virtual. Okay, so now what I'm going to ask you to do is to, if you already assembled some chromosomes and put the foam parts on the ends of your scaling graphic, go ahead and take them off and reconfigure those foam parts to now make these 4 chromosomes shown. And so the next slide, we're going to, as you're constructing these, the next slide will show these same 4 chromosomes, and we're going to go over some parts. So talk about them as you're constructing your chromosomes. That middle section that you see pinched in there, that is a centromere. So that's the region of DNA to which your spindle fibers and kinetic cores can attach there for organizing where your DNA goes during cell division. The tips, or the roundy tips at the ends, those are your telomeres or telomeres, as you know, telomeres or telomeres, potato, potato. Those are the regions of DNA that have repeating sequences to prevent or slow down the erosion of your DNA as replication is happening. Every time you copy your DNA, DNA gets shorter and shorter. So those telomeres help extend the life, so to speak, of your DNA strands. And then blue represents paternal chromosomes, the DNA you inherit from your father through his sperm cells. The red represents maternal chromosomes, the ones you inherit from your mom through her egg cells, through her egg cell. And for every single chromosome, it has its pair, so it's homolog. And so you can see there that the length of these chromosomes and the location of the centromere helps to determine which chromosomes are the homologs. So as you can see in these sample chromosomes, it doesn't matter exactly how you assemble them, but you want to have your students make a set of chromosomes that are a little bit shorter, some that are longer, and centromere position could be same in the middle or in slightly off center, just shown like the left chromosomes. So the reason that we're able to identify which chromosomes are which is based on a graphic that you're seeing here called a karyogram, and it's a way to organize visually chromosomes according to its size, so how long they are. So you can see pair 1 is the longest all the way down to 22, which is short and cute. And then you have your sex chromosomes X and Y at the very end. And you could see here there's 2 of every type of chromosome, and they have the same banding pattern and similar genes located on both of those chromosomes. The next slide will show you an actual photograph of chromosomes taken in the middle of cell division. So during mitosis is when we're going to take a photograph. So what you can see here is you have 2 chromosome 1s, and what's really important to let students know about is those chromosomes are in a replicated state. So we're going to talk about that in just a moment, but a lot of confusion happens thinking those 2 chromosomes you're seeing there, a lot of students may think that those are replicated, and that's why you have 2, but no, each individual piece that you're seeing there is already a replicated chromosome made of 2 sister chromatids. So let's go ahead and kind of talk a little bit about the cell cycle. So it's good to orient students. Earlier this week, yesterday, I did an overview of the cell cycle, talked about which types of cells divide, how often they divide, how old some of your cells are, and the life cycle of a cell. So it starts with G1, and then S is the synthesis phase where DNA is copied. But we can't really see visibly within, under the microscope, the chromosomes until we have mitosis. And so the next slide will show that, DNA replication is happening during the S phase. And then the karyotype is the picture of the cells taken during metaphase. So that's when the chromosomes have fully condensed, they're organized, lined up on that metaphase plate or equator of the cell. You can take a nice clean picture. So when we talk about these chromosomes and use them to model mitosis and meiosis, we want to distinguish the terminology. So in the next slide, what you're going to do is we're going to replicate our DNA. And so each one of the chromosomes you made, you're going to make an identical copy. So this is DNA replication. So if you guys can see my screen, I tell students we kind of actually do a physical calisthenics. I call it like bio exercise here. So I have students stand up and they put their arms together and they pretend they're a big long stick, and I have them say out loud, I am a chromosome. So this is a chromosome. And then they put their arms out and their legs out, and then they say, I'm a chromosome replicated. So with these foam parts, I have students hold them up and say, this is a chromosome, and this is a chromosome replicated. So both of those are called chromosomes. It's just the terminology they use, but this replicated chromosome is made up of 2 sister chromatids that are identical. So the same genetic information is in both sister chromatids. And so you're going to go ahead and do that using your foam pieces, so that as you see in the graphic, this is a chromosome with 2 sister chromatids, and here is its homologous chromosome made up of 2 sister chromatids as well. So you have the paternal chromosome and the maternal one. And this happens during the S phase of the cell cycle. All right, so if you've got that, hopefully you've got your original and then your duplicates, replicated chromosomes. Now we're going to go into the M phase of the cell cycle, which is mitosis. So mitosis is made up of prophase, metaphase, anaphase, telophase. And students are now, and you in the student role, are going to utilize your replicated chromosomes to model the different stages of mitosis. So in the next slide, kind of giving you a little prompting or hint for that as you follow along. And you can see Heather on her camera there. She's modeling it there so you guys can see, for those of you who may not have your kit in front of you. And so prophase, the chromosomes become visible. They attach to the spindle fibers. And so one of the things that's not showing in this diagram or included in your kit are these spindle fibers that you're seeing in yellow in this diagram below. So there's a couple of options. In the classroom kit, it actually comes with the kinetochores and the spindle fibers. And you can actually model the attachment of the spindle fibers to these sister chromatids and then show them being oriented in spindle fibers, helping these chromosomes to orient. So that's the classroom kit. Now, the at-home version, here's what I would recommend, is have students get some kind of string or dental floss. I've got some yarn here, and have them kind of lasso that chromatid and use string to model the spindle fibers attaching to the chromatids and then moving them around. And you can have them shorten and then pulling these chromosomes or moving them to the ends, the poles, like in anaphase. So one little tip or hint with what you might have students do as part of demonstrating their learning is you could have them take pictures of each of these phases and put them into some kind of document to submit with annotation. Or you could have them make a stop motion video with like the stop motion app. Or you could have them, if you're hybrid like me, in class model and walk through this. So one thing that's super helpful, I think, is if students talk out loud and try to explain to someone else what is going on. So in the classroom, I have them work in pairs. You could online have them do breakout rooms where they're in pairs and they model on camera and show their partner what's going on. Or if they're solo kids at home, you could just say, find your dog and teach your dog how the process of mitosis works. So often a dog is a good best friend and helps with learning science. So those are a few recommendations I have. And then you might also have them, if they're able to be in a classroom or maybe at home, to get those table markers and window markers and be able to draw spindle fibers, or also where the nucleus is, and maybe erase as well. So maybe on a whiteboard. So those are some different ways of being able to show that either in the class or at home. Okay, so the next video is just an animation to show the classroom kit, which actually has 3 sets or a total of 6 chromosomes there. And you could see them being lined up in metaphase. Those are the replicated chromosomes, spindle fibers attach, and you have the bundles of 2 sets of DNA in 2 new cells. Okay, so as we go through mitosis, I asked my students several questions to see how their understanding of daughter cells versus the parent cell, how many chromosomes are each cell, and how many chromatids. So the language is super confusing for students with them understanding centromere, chromatid, chromatin versus chromosomes. So it's really important to clarify the language that your students use and have them, as much as possible, verbalize out loud with their own lips some of these different terms so that they can demonstrate understanding. Now we're going to go into the other type of cell division, which is meiosis. One thing I do, and this is really weird for me to say meiosis, because in class, I mispronounce it on purpose. So I usually tell students, if you want to make more of your body cells, like my toe cells, you're going to use mitosis. And if you want to make a new baby, a little mini me, you're going to use the cell division called meiosis. So I usually call it mitosis and meiosis, and I make sure that they know most people call it meiosis, but I like to say mini-me's, meiosis. So this is the type of cell division done by egg and sperm. And the whole purpose is to do reduction division so that for human cells, the number of chromosomes gets reduced in half from a diploid number of 46 down to a haploid number of 23. And one of the things that this kit can do, we're going to see in just a moment. So the next slide, we're meiosis or meiosis starts off similar to mitosis, where you have the chromosomes in the G1 phase are going to be singular. Okay, so they're not replicated. So these are chromosomes. So you're going to go back and have that as singular chromosomes right there. Okay, and then in the S phase, you're going to have replicated chromosomes, as you see in that picture. So that's going to start off the same, mitosis and meiosis. And then we're going to get to the special part of meiosis, which is introducing the idea of crossing over. So meiotic crossing over is a fairly complex process, and it's where these homologous chromosomes come together and they end up joining to each other. And you can see that where they join together is called a holiday junction. I don't know the ins and outs of all this. It's fairly complex, but the main point of this crossing over is so those homologous chromosomes can be connected together and stay together as they go through that first division, so we assure that homologous chromosomes are paired together. And so there's going to be breakages and joinings happening with your chromosomes. So this kit is great. So in the next slide, we're going to actually model meiotic crossing over. You can see in this animation with the classroom kit, you can have the nucleotides. This is an animation showing meiotic crossing over at the nucleotide level. So this is a great video to show in terms of having students see how those homologous chromosomes are attached together. But what we have here is our large foam chromosomes. And so we're going to model prophase 1. The two main processes you're going to have is synapsis, which is the coming together of the homologous chromosomes, where you have 4 sister chromatids all grouped together called a tetrad. Now, one of the things that is misleading about this picture, as we talk about how all models are useful, but all models are wrong. In terms of this orientation in this photo is showing chromosomes in 2D space. And what you may want to have your students do with this is orient these tetrads so they're in 3D space and they can actually sit on top of each other. And so you may want to do that with your chromosomes. And so that when you see crossing over happening, it could be, if you can see here, where you kind of take this one piece and take off a part and swap it. And so in this case, you could swap it like that. So you're seeing that crossing over happening. Now in the picture, you're seeing if you had 2 homologous sister chromatids next to each other, maybe I should do it that way, you can detach pieces and swap that. So in reality, there will be 1 to 3 crossover events per chromosome. So you can line your different homologous pairs together. And I'm just going to pick maybe the tips of this one. You can pick what you want. It's random, and that students can choose that. And then you can have some different variations there that are happening over. Okay, so that's crossing over. One of the things I love about this kit is that we have the ability to demonstrate this crossing over and the detachment of these pieces. And then what's fun with high school students especially is they're not always listening to directions. So what can happen is they're trying to do crossing over, and I've had students end up with something crazy like this. Let me just show you briefly. And then they're like, Mrs. Chou, look what I did. what happened? And I'm like, great, you just had not, you know, crossing over and mutations. And that could be an example of something that, you know, could happen in class and in real life as we look at these inversions or the translocations for this one of different chromosome pieces because of crossing over mishaps happening. Okay, so moving on, I want you to show crossing over and then continue on and demonstrate the two cell divisions, meiosis 1 and meiosis 2. And we have there we go. So as you go through the two different divisions, meiosis 1 and meiosis 2, you can see that in metaphase 1, you have the independent assortment happening there. And so between maternal and paternal chromosomes pairing up at the equator, there's different ways that you can pair them up right there. And then already at telophase 1, you have half the number of chromosomes, 2 instead of 4. And as you continue on, you end up with 4 different daughter cells in telophase 2. So let's go ahead and take a look at the video that will show this process with 3 pairs or 6 chromosomes. This is like the classroom kit. And you can see the spindle fibers attaching to these chromosomes. And again, your students can use some kind of string, or if they're doing it on a whiteboard, they can draw those spindle fibers out to show their attachment and the orientation of your chromosomes. Okay, so at the end of the activity, it's important to ask students about the chromosome number. At the beginning of meiosis, the end of meiosis, where are cells diploid, and how do they end up becoming haploid or having half the genetic material at the very end of this process. And then going back to mitosis, asking students to show maybe having in class, you know, simultaneously mitosis versus meiosis, comparing how do they differ, and then how are they similar to each other. I think these kits are really powerful because as in the classroom, or perhaps you're watching them on video online, or they're by their photos documenting their work, you can see where student misconceptions lie and where the crossing over is done weird, or they're lining individual replicated chromosomes along the metaphase plate versus homologous pairs of chromosomes. And so I think this is where the kit is really useful to reveal student misconceptions. And of course, as students manipulate these chromosomes, you can talk about the variations where variations occur, crossing over with the independent assortment of chromosomes in anaphase 1, and then random fertilization of gametes. So having separate different kinds of combinations of maternal and paternal chromosomes that end up in your gametes there. Okay, so the final thing what we want to do is to be able to talk about how you can use this kit to connect it to genetics. So you can ask students to play the role of a genetic counselor, and there's a young couple that's going to come to you for some help. And what they're going to be concerned about is a condition that involves the beta hemoglobin gene, the beta globin gene. I see here there's a question coming in from, is it Louisa? Are there interactive websites to achieve the same effect? I am not sure about the interactive websites. I think that clay could be used, like Play-Doh. There's also the student modeling activities that Diane just put up as a reference. That one is helpful because you can have students drag and drop chromosomes. So definitely you can use that. and then be able to show like the independent assortment of chromosomes through this digital resource. So there's a Google Slides or PowerPoint version of that that your students can use digitally. I think it's really important for kids to show modeling in multiple ways. So I think the online version versus, and also physically in some way. I've also used before like socks. You may have seen that before as a product, or pipe cleaners. Now, I think the disadvantage of that is that students can show, you know, the replication of chromosomes and orienting them on the metaphase plate and anaphase and such, so forth and so on, but you really can't show crossing over very well. So I think that's where this kit or something like Play-Doh might be really useful for students. But my students get bothered when they mix Play-Doh. So this, you won't have to worry about mixing the Play-Doh. So as we take a look at the role of a genetic counselor, one way to model a condition that commonly occurs is looking at the beta globin gene. And so the beta globin gene codes for the beta globin protein, which is one of the subunits of the hemoglobin protein right there. So there's 2 alpha globins and 2 beta globins. And within the middle of each of these subunits is a heme group, which contains iron. So this protein is found in red blood cells in abundance, and its purpose is to aid in carrying oxygen for cells. So let's take a look at the coding for this gene, where it's located. The beta globin gene is located on the short arm of chromosome 11. And for students, you can remind them that these large, big, fat chromosomes actually contain hundreds or thousands of genes, and each gene has a location. So I think that's important to point out. So you could use maybe a little piece of tape or something here to indicate where this gene is located. And then as students zoom in to the molecular level of the nucleotides, you can remind them of this scaling graphic with the nucleotides in the middle, and have students pull their nucleotides out, and you have them actually construct a 3 nucleotide sequence of one of the loci or of this particular gene. So we're going to zoom into one location that is the most troublesome one. And so using your nucleotides now, you can model this little segment of DNA in this gene. And you have to model 2 copies of them, because you'll have 1 copy on the maternal chromosome and 1 copy on the paternal one. The 2 different variants that we're going to take a look at is the one on the left is the big H allele, which will code for a normal beta globin protein. So they call that hemoglobin A. That's our wild type human hemoglobin. And then the little h allele, as you relate it to genetics and using abbreviations, codes for the abnormal beta globin protein. So with this, you can ask students at this point, what looks different between the 2? And do you think that one little difference in DNA will make a big difference with the phenotype and with this protein, and then the expression and how it plays out in the health of the cell? So in the next slide, you'll see that one little change, which is a substitution from that T down there to an A, changes the RNA sequence, and that codon, which was a GAG coded for glutamic acid, now codes GUG, which is a valine. So let's see in the next slide what that might lead to. So in glutamic acid is a hydrophilic amino acid, and its normal position, you're seeing it on the left, is on the outer part of the protein. It loves water, and so it allows this protein, based on its location and its chemical properties, to be able to fold up into a globular shape and form the 4 subunit piece of hemoglobin. Now, changing that glutamic acid to valine. Valine is a hydrophobic amino acid, and that means that it causes the folding of the beta globin protein to change. And you end up having these hydrophobic pockets sticking out at the end that attracts other also abnormally shaped hemoglobin molecules, and they start to clump together and form a long string of them, like the picture shown in the right. So the change in one little nucleotide causes a change in the amino acid and the change in the tertiary structure folding of this protein, which changes its chemical properties and how it connects to the other molecules around it. And so letting students know that, let's go back to taking a look at what possibilities, next slide, of that you could inherit. And so you could inherit 2 normal copies of the beta globin gene. And so that would be the first one. And you can inherit 1 copy of the normal, 1 copy of the abnormal. And so that would give you a genotype that would be big H, little h. And in that case, we say that individual is a carrier or has the sickle cell trait. Now the bottom option is you could inherit 2 mutated copies of the beta globin gene with that particular nucleotide sequences. And so the genotype is little h, and unfortunately that can lead to sickle cell disease. So sickle cell disease means that all of the proteins that are made, all of the beta globin proteins that are made will have that sticky property and will clump together and form those rods and start to deform the shape of those red blood cells. So instead of the nice little round red blood cells, you're going to have the sickle-shaped ones. And through the capillary, those round blood cells flow through perfectly, but the sickle-shaped ones will create clots and other kinds of problems. So lots of different health issues associated with having sickle cell disease. So this couple who comes to you, because you're an expert in genetics and understanding the inheritance of some of these traits, this young couple, the Smiths, want to start a family, and they know there's a history of sickle cell disease in their family. And so they get tested and find out they're both carriers. So they both have a normal copy and an abnormal copy. And so now you can have students model out the potential outcomes that their kids might inherit. And so going back to the nucleotides, have students use those nucleotides to put on a Punnett square. So in the kits, we have the paper copy of this Punnett square, and you can have students set those nucleotide sequences representing big H and little h, normal and abnormal hemoglobin, and then create different combinations. So the student kits don't have enough of these to actually create all the possibilities, but you can have them move the parental alleles. And so if you click the graphic or the animation there we go. You can have them show that in a video. You can have students take pictures of each one of these boxes to show the results. And what's great is the bottom left box that shows little h, big H. A lot of students are confused by that. And you can just say, well, you can reorient the letters so that the big one shows first, but that's what alleles are inherited from the parents. And so if you can see there, so the kids are going to have a 1 in 4 chance of inheriting a normal phenotype, 2 in 4 chance of being having sickle cell trait, and then 1 in 4 chance of having sickle cell disease. So you can see there, and that's the probability. And so as a genetic counselor, you would counsel that young couple on their options and outcomes and helping them to make informed decisions based on what they know. So this kit can be utilized for a variety of things, all the way from cell cycle to mitosis and meiosis, looking at the DNA level, maybe even talking about replication, and also genetics. So many different topics that can be covered with this kit that can be used in the classroom or at home. So the next slide shows some connections with the NGSS and the standards covered. And the very last slide, let's go ahead and do there, shows the code for the modeling kits, a coupon code to receive a discount, and where the lending library is too. So that's a link there that's helpful. So some of you are in the position of you don't have the budget to purchase all of these kits for your students. You can borrow them. They will send it to you if you have the funds to pay for the shipping to send it back. So I think that's a great deal. It's certainly a lot cheaper than buying the whole thing, and a fabulous way to be able to provide that resource for your students as well. All right, so we have a few minutes left, and I would love to answer any questions you have or receive any feedback.